Machine learning for transcriptomic data, from raw reads to data exploration, machine learning, and visualization. This is a preview of a workshop that we did at University of Nebraska in Omaha on May 15th.
The T-Bioinfo platform was designed specifically for research use, but we're using it for education. We're building a full curriculum around the platform. As you can see, this is the areas of analysis that shows different types of data that could be processed, integrated, and analyzed on the platform. Today, we'll be covering this section called transcriptomics. There on top, you can see RNA-seq. You can probably also see this on your screen. And this section allows processing and basic analysis, things like differential gene expression. Now, the second section over there on the bottom is data mining, and there we have unsupervised and supervised machine learning methods. So those methods include things like principal component analysis, clustering, and for supervised classification, things like SVM. The third part is annotation. So once you have those genes, how do you annotate them in terms of differential expression on pathways, as well as how do you just get from ensemble IDs to gene names and functional annotation. So we'll look at how to generate these pathway maps and how to look at annotation on the output data. The type of data that we will be using today is around modeling of precision medicine. So basically we'll use a cell line data set that was used to investigate breast cancer and response to treatment. So why are we looking at this data set? So we have multiple types of clinical subtypes of cancer. For example, this is a mammogram. You can see here this white area, and that supposedly is a tumor. And now the question is, how do we classify? So one of the challenges is to identify the source of the tumor. So which tissue is it coming from? And one other way to look at this is what proteins are expressed in that tissue. So we can do immunohistochemistry, but the limitation is that, as you can see here, we only have a small number of genes that we can actually look at, small number of proteins. So then we moved on, and now we have array data, and array data helps us to look at gene expression and classify different subtypes of cancer, in this case, breast cancer, by gene expression. So you can see that the red is going to be high expression, green is going to be low expression, and then we can see the different subtypes are expressing different genes differently. Why is this important? So first of all, it's important to predict survival. You can see that here a survival plot or a recurrence plot that shows how this panel that we just saw for microarrays separates different subtypes of cancer in terms of their survival over a number of years. And you can see a comparison of two panels here, Oncotype DX and PAM50. PAM50 is something that we're going to be discussing today. The same approach is used to select treatment. So by looking at the expression, we not only know the prognosis, but we also can select more aggressive, less aggressive treatment or combinations of treatment. And finally, we can also start looking at targeted cancer therapy, right? So how to not select a generally toxic treatment that kills some cell division, but also select something that interacts with a specific pathway. So obviously we need to select which pathway that is. So this is a broad overview of a publication. This is the publication. You have the link, and you can download all of this data set. It's publicly available data set. It combines multiple types of omics data and response to treatment. So it actually measures half-time of complete elimination of division of these specific cancer cells. Now, all of these files are already prepared for you. So let's make sure that you have all of the files that we'll be working with. There was a download file button on the page. So let's find this file called samples.svl. It's going to be under input files, under expression quantification. If you open that file, in WordPad, it looks something like this. Okay, so this is what the file looks like inside. What does this mean? This is just a reference. So instead of uploading multiple FASTQ files, which are large, we're going to take a number of references of files that are already pre-uploaded on the server. Okay, the second thing that you will notice here is that each file has a _1, _2.fq. So these are pair-end files. Most of the things that we are going to be discussing today also are available online. These are courses called Transcriptomics 1, 2, and 3. They cover all the basics of working with RNA-seq data. So the first part of our presentation is going to be around sequencing and why it's important to select different approaches to prepare the data for machine learning. So as you know, there's a genome, and then we have a gene A and gene B. And those genes are going to be, some are going to be expressed, some are not going to be expressed, right? So when we have the sequencing data, we're looking at reads, and those reads represent multiple genes that are being expressed. Now, in reality, this is a little bit more complicated. So there is going to be shattering of those reads, and then there's going to be adapters to those reads, and then there's going to be PCR amplification, and then finally we will have the reading procedure. So whenever we look at raw reads, there's a little bit of a complication there. And therefore, whenever we do the RNA-seq analysis, it actually consists of 3 different steps. Step 1 is pre-processing of the data, and notice the colors. The colors are going to correspond to what you will see on the platform, just so that visually you will be able to spot those different methods. So pre-processing, adapters removal and additional trimming, and then removing the PCR duplicates. Mapping, which is mapping on a set of known transcripts, so genes that are known, and identification of novel transcripts like isoforms. And then we will also discuss the combined strategy. And finally, quantification. How do we quantify the alignment of reads? So this is the basic pipeline. Okay, so if we go to the platform, if everybody has the platform, you can go, okay, RNA-seq. If we click through, just to quickly explain to you how to do this, if you can do this together with me, you'll see that this is a very simple and quick thing to do. So first of all, you'll see a selection here on top. We will be using our model genome. Here, organism, we will select Homo sapiens GRCH38. FASTQ files over here, and then over here we will use pair-end, so PE. As you remember, we have the SVL file. So instead of uploading individual files, we will actually go to URL bulk upload. Okay, and here we will use the SVL file that you already have, .svl, samples.svl. It's a small file, so it should upload really fast. All right, so now that we have the file uploaded, if you click on continue or prepare groups, you will see that the list of files is on your screen, correct? Do you see this? And since we're not doing differential gene expression, we don't need any groups, so we can do cancel, no groups. Now we're ready to build our pipeline. So you can see that the buttons that we can select are darkened out. So if you click on start, which is the only button that you should see darkened, now you have a limited selection. Remember the colors. So what are the colors? We see blue over here. Pre-processing is blue. You can see the red ones. Those are the mapping sections. Okay? And then you will see this one will actually be green, RSM expression table, expression levels. So let's start with Trim-O-Matic. Trim-O-Matic is going to trim those adapters. So we're going to choose that. Then we're going to do PCR clean. This will use an algorithm for selecting duplicates that are not related to the biological representation of additional reads. And now you can see that the blue section is complete. We have the pre-processing steps right here. Then we're actually not going to touch anything over here. We're going to jump over to Bowtie2T, which is mapping on known transcripts. So we're not interested in new isoforms, in gene fusion, in any kind of variation in the genome. We're just going to map onto a known GTF. And finally, you can see that this is red. And then finally, RSEM expression table. Let's click on that. And what you'll see here is a number of selections that we will use to quantify the number of reads, right? So what are these values? We'll talk about this in a second. In the meantime, let's just do FPKM, and let's click on it. Now you can give your pipeline a name. You can save it. Now you can run it on your account. I'm not going to run it because it will take some time, and I already have the prepared output file, so I'm not going to be running this right now. But what you will notice is under My Pipelines, you can see the pipeline that I've created. Now, if we return to the presentation, so this is the pipeline that we built. We built this pipeline, and our goal is to get an expression table. If you look over here, this is directions for you to create a new pipeline. We're not going to spend too much time on this because we don't have that much time, but let's do it for practice. If you can follow along, you completed the previous steps, everything went well, let's try and do this. All right, so this is what your pipeline should look like by now. And here you can see expression quantification, a folder called expression quantification. Now this is important to understand because before we can analyze any kind of table using advanced machine learning techniques, we actually want to understand what are we looking at? What are the features? So what are the rows or columns that we have? And what are the numbers in those cells? So let's take a look at these two tables. So what you'll see is you actually have two different tables, one called expression genes FPKM, or not filter, and one called expression genes FPKM. So if we go back over here, this is what this looks like. You can also open the file in Excel. So let's open this file in Excel. I'm using a Mac here, so you might be seeing something a little bit different, but basically any table manipulation software is going to work, unless it's made by Microsoft. Now let's take a look really quickly. What are these IDs over here on the side, right? So these are gene names. These are ensemble gene names, and we can scroll through and we will see that there's a whole lot of them. So go all the way to the bottom. There's something like 20,000 genes here. What are the columns? So let's go to the very top. And in the columns, you can see that the name of the sample is going to be the name of the column. So basically, we're looking at expression of an individual gene across multiple samples. Okay? Now let's go back to our presentation and take a look over here. Gene ID. So if I have a gene like P53, for example, expressed over different samples, I'm interested in the variation, how those numbers change across multiple samples. That will give me some insight into how these samples are different by their expression values. Now, can you select the second gene in your table and the second column, so the third column from the beginning or the second sample, and tell me what is this number? What is the number that you see in that cell? I'm using
filtered one.
Filtered. Okay, so to understand what this number is, I hope you found it, right? So you remember what that number looks like. But let's take a look at what it means. So we have a number of ways to quantify the number of reads that were aligned to that segment. And here we're looking at the FPKM value. So FPKM and TPM are two methods of normalizing those values against the raw counts. That is also one of the options that was available in the settings that we had. The raw counts are just looking at the number of those fragments repeated. But FPKM and TPM actually normalize it for the length of the gene. So if you think of a very long gene and a very short gene, the number of repeats of those genes is going to mean different things. So we have to normalize it against some universal value, and that's 1,000,000 reads, right? So you can see fragments per kilobase per million map reads. So those are the two ways of normalization. We're not going to go too much into detail here, just for the sake of time, but just to explain what it means. The second way to normalize these values is to use logarithmic scale. So if you remember, the logarithmic scale basically reduces the numbers in such a way that very low and very high numbers are going to become closer together. So instead of looking at huge variation, we are looking at variation that is normalized. Now, let's review this really quickly before we proceed to the next stage. The first part that we did was pre-processing. So we used Trim-O-Matic and PCR Clean. Trim-O-Matic reduced the adapters. PCR Clean reduced the amplification that PCR does. The second step that we did was mapping. We used a mapping algorithm that put the reads on a specific one is we quantified those numbers. So instead of just looking at the reads, we used a complicated formula and generated values in FPKM. Now, does this even matter? A lot of people have a question whether this matters, the pre-processing, the Trim-O-Matic and PCR Clean. We could select a raw table without any pre-processing, and we could do a table with the pre-processing, which is what we did, right? You remember in the names of our samples, it actually says that it's pre-processed, right? So here we did a comparison, and basically in this comparison, what you can see is in the upper corner where the values of expression are high, you will see that the values are going to be skewed if we don't do the pre-processing steps. Okay, that's just to say that whenever we deal with gene expression, those pre-processing steps are going to affect primarily those genes that are going to be highly expressed. And why is that important? Because highly expressed genes is typically what we're going to be looking at. All right, so quick overview of RNA-seq. I hope it wasn't too basic, but if it was, this was just to bring everybody up to speed and so that we all understand what data we're working with. We do have a few previous workshop questions that came up. So if you just look at the pipeline, you'll see that there are other sections and other buttons that we didn't cover. Error correction, different types of mappers, and other sections doing differential gene expression and segmentation. What are those? In 2 words, error correction is primarily used for microarray data or older sequencers. Whenever the older sequencers were used, the quality of the quantification wasn't very good. You had a lot of base pairs that were replaced, and so these error correction methods reduced those the bias that came as a result of that. Different mappers, what's the significance of different mappers? Different mappers use different algorithms, and they could be useful in specific situations. So some papers that you will read use star, some paper use top hat, but basically it's your preference. There are some more updated and less updated ones. That's the choice that you have to make. Differential expression, we have a number of different options for differential expression. All of them function in a very similar way, but again, there are some more updated ones. And then segmentation. So segmentation is a separate section that requires its own explanation, just to let you know that it's available there. And once you do segmentation, you're looking for completely new segments that are being expressed. Sometimes it's useful to annotate those using BLASTX. So you'll see that section there as well. All right, now part 2, I'm going to give it over to Stepan here, and he's going to walk you through how to use this table and use machine learning to identify patterns in this table.
So first, let's start from definition of machine learning. Actually, machine learning is separated into 2 huge categories. One is unsupervised machine learning, and second is supervised machine learning. Let's begin from unsupervised machine learning, and suppose that we have a set of images. And actually, we don't know is this dog or is this cat? So we have unmarked images. And the task of unsupervised machine learning is to split these images, not marked images, into several categories. For example, here we can split it into two categories, cats and dogs. So in machine learning, we have unsupervised machine learning, we have unmarked samples, and we want to mark them if it's possible. Okay, we can show these samples on the plot, on the scatter plot, and actually we can have outliers here, and our algorithm can make errors. This is not prohibited. Every algorithm can make error. Let's return to our data. In our data, we have some samples and some genes. So our columns are samples and our rows are gene names. And we want to apply unsupervised analysis methods to our samples. So we want to see if there are any division of our samples into some groups, which can be, for example, biologically meaningful. For example, different cancer types or something else. And let's take a look for first method, principal component analysis. This is just a method for visualizing the data. For example, imagine that we have only two genes. So each sample is associated with two numbers, expression of the first gene and expression of the second gene. In this situation, we can make just scatter plot, yeah? We have two numbers at each sample, and we can draw it and see that this is the first group and this second group. But actually, we have a 7,000 genes or more, and we can draw it in our plane. So it is points in 7,000 dimensional space. So principal component analysis is just a method for optimal projection of high dimensional data to the plane. So, for example, imagine we have a pen, three-dimensional object, and I project it to some plane. So if I will project in this way, I will see only one point here, yeah? And I can understand that this is a pen by this projection. And if I will project in this way, I will see actually a pen. So not each projection is good. So principal component analysis is an optimal projection, which maximizes information which we can obtain from this projection. So how to use it? Let's go to our platform. Okay, and let's click upload files. Here our files are not huge, so we can just click add files and go to the input file section. Here we have unsupervised 7,000 genes folder, and let's select cell lines expression data.txt, not marked one. Choose, then click continue, then start. Then we select PCA draw, not PCA draw to have a plot. Here we use default options, save, and go to the end. We'll assign some name to our pipeline. PCA. Oh, no. Just simple PCA. Save. And then we can run this pipeline. So if I click run pipeline, I need to run it. And then I can go to the My Pipeline section and see that state of this pipeline is started. So it's running now. And as we said, we prepared output files folder, and select here unsupervised PCA 7,000 genes not marked. And here we see output of our pipeline. So it contains one PDF. We can open it and see our samples on the plane. So it's just the optimal projection of our 7,000 dimensional data to the plane. And here we see two groups. Yep. So this is the first group, and this is the second group. Just some natural division. And let me return to the presentation. Actually, we work with cancer cell lines data. So we have here 52 samples. And actually, these 52 samples are already grouped to the 4 categories, 4 different types of cancer. And we can draw same plot by assigning colors to these groups. Here we see 4 groups. Yep. This one type of cancer, second, third, and one more. So actually, we've seen only a division to 2 groups, but actually there are 4 groups. And here is some important idea that we have 7,000 genes. It's a huge number, and we have a lot of information in that. But we have a lot of noise with these 7,000 genes. And the idea is that if I will reduce number of genes, so select some features, my plot can be, my data can be plotted better. So for example, if I will take 50 genes from PAM50 panel, my plot became better. Here you see some 3 groups. Actually, we don't see 4 groups because PAM50 panel is not aimed to our 4 groups, but here are 2 groups which are highlighted. Okay, here groups. And let's go to the next unsupervised analysis method called hierarchical clustering. So first method is just a method of visualizing data, and this hierarchical clustering is the method of clustering the data. So let us create the pipeline. Go to the unsupervised analysis section. Upload upload same file. Press continue, start. Here we have hclust button, and we need to change some options here. For example, we see linkage is default to single. We need to change it to ward.d2. I will explain what it means. And we expect, for example, 4 clusters here. And then press save and go to the end. Assign some name to the pipeline. hclust. Save. And then we can run this pipeline. Okay, it is running now, so not to waste much time. You can see already results in your output files folder, hierarchical clustering 7,000 genes, and here we see 2 PDFs. Oh. Okay, let me explain the idea of hierarchical clustering first, and then look to these PDFs. Imagine we have points on the plane. In our case, these are points in high-dimensional space, but imagine that it's points on the plane. And at the first iteration, we assume that each point is a cluster. So we have 1 element clusters here. At the second stage, I found 2 nearest clusters. So our clusters is 1 element, and we found 2 nearest points. And then we merge these 2 clusters. So now I have 1 2-element cluster, and our other ones are 1 element clusters. And then I repeat this procedure. Then I find new 2 nearest clusters, for example, 2, and so on. For example, on this on this stage, we merge these two. And at the end of the procedure, we have one huge cluster, all points. And this procedure can be visualized by plotting a dendrogram. What is it? It's a tree. Height of the tree is a distance between points. So rate of dissimilarity, for example, if height is low, so at the bottom, that means that samples are really close to each other. And if it is high, it means that they are far enough. And on this plot, we see, one second. I don't. Okay. Here we see 4 clusters. For example, this is the first cluster because these points are similar, and they are not similar to other ones. Here's a big second cluster. There's a third cluster, and there's a fourth cluster. And we can cut this tree to mark our samples. For example, we can cut our tree in this place. So this line intersects our tree at 2 positions. So this is going to be first cluster and this second. We can cut from this place. So first, second, third, and fourth. Okay, and now a little practice. You see that we have sample names that are leaves, but also we have file in where's my mouse? In input files directory. Let's go to it. Input files, 7,000 genes, expression data marked. Let us open it in Excel. Right. Okay, or here, and we see that here's a header. It's already modified.
So if you look at your data,
Okay.
now before that we had both lines. We had sample IDs and the group names, but now we're just having one line, and so you just use the file as is and run the H clustering pipeline that looks something like this. Yeah.
Here's a tip. So we see that our division is actually correct. Here we see one group, there's a huge subtree, the second small one, and yeah. So we see that basal group is actually very similar to normal life group. They are shuffled. Now about supervised machine learning. We have a really different concept here. Here we have training set, just images with labels. So they are already marked by somebody. And then we have a test set, some pictures, some samples, which are unknown. And we want to train our classifier on training set and then make some prediction on test set. So we want to assign a label to each picture in the test set. So in our expression tables, it means that we have some samples with known clinical cancer subtype, and now new sample arrives, and we want to assign a clinical subtype to it. Okay, and here we have some methods for doing that, and let's start from stepwise linear discriminant analysis. Stepwise LDA, we can run it by going to the supervised analysis section. Okay, let me show the input files first. They are in input files folder. Here we have supervised 7,000 genes, and here we have 2 files. First is called train, and second test. Training set, we have labels. So we are sample names, and here are group names. So they are marked. And the training set, oh, how much? The training set contain unmarked samples. So we don't have group names here. And our task is to mark these samples. We just need to upload these two files to the platform. Add files. Input files, supervised 7000, train, and test. So we upload both of them, then click continue, then start, stepwise LDA, and we need to modify this statistical threshold. Just add some 0 and change 5 to 9. This experimentally estimated threshold, so hyperparameter for the algorithm. Then save, and then end. And then we can run pipeline. This pipeline will take about 5 minutes. And how did I obtain these two training and test files? I just taken randomly one part from huge table as training set and the other part as test set. So actually I know real classes for test set. So let's take a look at output. Output files, supervised, stepwise LDA, and here we have file called prediction test. Let us open it, and here we see that classifier assigned group name to each sample. So as I said, I know real classes, and classifier made one error on this sample. So actually it's normal-like, and it said that this is basal. So this is normal for classifiers, so we can't achieve 100% accuracy. And why we selected stepwise LDA, we have some bunch of methods, because stepwise LDA performs feature selection. Here we see stepwise feature coefficients.txt. We can open it. And here we see some gene names. What is these genes? These genes are marked as informative by the stepwise LDA. So it used them for classification procedure. So we just can take these genes. They are located at input files, annotation, 15 genes file. And these are highly informative genes. Do we have time for it? Okay. So then we can use these 15 genes for another methods of classification, for example, for support vector machine. So we will not speak today about this, but idea of feature selection is very important because many supervised machine learning algorithms can't work when we have really big dimensions. So when number of supervised analysis algorithm can't be applied in this situation. So for applying them, we should select features first. And stepwise LDA or random forest classifier are our friends in this task. So they perform feature selection. Okay, that's about it. And then let's look how feature selection can affect on unsupervised analysis. So now, when we have 15 informative genes, we can rerun our PCA with only these 15 genes. And here we see that uh, the plot really looks better. So we removed all noise, and PSA plot looks really pretty. And here we see 4 groups. Remember, last plot only 2 groups, and here we see all the groups. And so hierarchical clustering also looks better now for clusters. Yeah. Yes. And now basal and normal like are not shuffled as in the previous case. Okay, that's about supervised analysis.
All right, so the main question now that we're going to be working with is we have these 15 genes. What are they? How can we understand what pathways they belong to? What could they functionally do? So you have this table of 15 genes, and we're going to create a quick little pipeline under the utilities section that will help us annotate those gene in ensemble IDs to actual gene names. So transfer this table to what you see on your far right. Okay, and here we go to areas of analysis, utilities, upload the file, so we can add the file. And here, by the way, you have a couple of ways of doing this. If you're interested in keeping your gene expression, you can just take the input file, annotation 15. You can take this one, or you can also take this one with expression values. In our case, let's just take the simple table so that we see everything close together. Click on Start, Annotation, and here take a look at the settings. So you have what databases do we have here represented. So we have the ensemble. So ENS at the beginning of the gene ID is ensemble. And so we have this database. And also we want to do gene ontology categories, right? So we can select yes or no. Again, we just want to specify that we do have a header, that we have sample names, and number of columns that it starts with. So which column are the ENSG names in? Column number 1, right? So let's save that to end. Run pipeline. Now, as you can see in the result, once you've done your pipeline, you will get something that looks like this. A gene ID, gene symbol, gene description, gene ontology domain, category, etc. So what we can see here are 2 good examples, DCN and EPCAM. Now, these are files that were the black box selected by stepwise LDA, and we want to see if there's any relevance of these genes to what we see in cancer biology. So first of all, remember, this is the heat map that we built, right? And here we can see those 2 genes highlighted as well. So the first gene that has green in the first samples and kind of, goes to green again in other samples. So it's not really present in a lot of these samples. And the one towards the bottom, some of it is highly expressed across the samples. But in our case, let's take a comparison of these 2 groups. So we will compare cloud and low and normal light. So instead of looking at everything, we will just look at 2 small groups. Okay, so what are these 2 genes? One of the genes is actually a known oncogene called decorin. DCN stands for decorin. You can see here a couple of publications on this. Oh, and we excluded the slide that actually shows the variation. But let's quickly take a look to make sure that this variation does make sense. Okay. So tell me if you have the annotated file ready. Let's take a look. Okay, you see mine is done. I'm going to just go down here, 15 genes out. And I can see that the first gene, 11465, is DCN. Okay, so this one is the gene of interest. And now I'm doing interpretation part of the work. So I want to take this gene, and let's just see its variation across all the samples. So what can we see? Okay. What can we see here? We can see here, well, let's expand it a little bit more. Okay, you can see that all the Claudin low are highly expressed, right? This one, this one, this one, but apparently not all of them, right? So 2 of the Claudin low are not highly expressed. There are some basal that are already also highly expressed, and then it seems like all the rest, the luminal, most of the basal are low expressed, right? So if we go back to the presentation and we take a look at these publications here, we can see what they're related to. So the presentations tell us that one is extracellular matrix and tumor microenvironment. Another one talks about prevention of metastatic spreading of breast cancer. What do you think this might indicate for the Claudin low with highly expressed gene? But what do you think they are related to? What does this suggest? So is metastatic good or bad? Bad, right? So now we've identified some quality of those tumors. Might suggest that one is a little bit more aggressive, another one is less aggressive. Which one is more aggressive? The one where it's high expressed or low expressed? Highly expressed? You're saying highly expressed is bad. Is that what you're saying? So DCN, decorin, is an oncogene, right? In terms of oncogene, it's related to cancer. We're not going to call it oncogene. Typically means it drives cancer progress. But in our case, it's a gene related to a cancer, right? So, but it's high expression. Is it good or is it bad in the patient? Bad. High expression is bad. Well, let's look over here, right?
Title.
It says decorin prevents metastatic spreading of breast cancer. So what do you think? Is it good? Right. So it's like a tumor. So it's not a tumor suppressor, but it prevents metastatic events, right? So maybe high expression, if we detect high expression, it makes the cancer a little bit less dangerous. What could this imply for the treatment of that breast cancer? So in cancer, one of the primary goals in early detection and treatment of cancer is to stop metastasis, stop it from spreading to other tissues and across the body, right? Because when it's localized, we have simple methods of dealing with it. What are those methods? So yeah, we can cut it out or we can do radiotherapy, right? So those are the 2 basic ways of doing it. And why is chemotherapy more dangerous, for example? Well, because chemotherapy is going to be given to the whole body, right? It's going to go everywhere and potentially or likely even cause other cancers, right? So now we have an insight, right? It's not anything proven, but at least it puts us on a track towards some biomedical application, right? So here we have, what we're trying to paint the picture is that we had some raw data. Nobody told us how to generate this raw data. It was a machine, right? A machine just generated 20, 30,000 genes. We looked at the genes, knew nothing about it, and then we ended up with 15 genes that actually make some sense in terms of applying that information towards further investigation or potentially in the long run, if it's confirmed, using it for some method of treatment. Now, before we go on, and stop me and just tell me, Sahil, also if there's anybody online, we have prepared projects like this one with the cell lines that go through the full data set. So here we're talking about a small subset of the samples. We take only one type of data. In the project here, it goes through the genomic data and uses multi-omics integration to actually work through what the paper discovered in terms of assignment of specific treatments. But we also have other types of projects here: analysis of the changing immune response in cancer, whole transcriptome profiling of cancer tumors in mouse PDX models, and even a TCGA, so a full pan-cancer type analysis. Homework. Whenever you have time, nobody's going to grade you on this, but for you to try it out, you can try the PCA plot with the 15 genes. We showed you the picture. If you want to try and do this at home, you've got all the data. If you run PCA, see what you do, what you get, and also genes from the differential expression analysis.
So that's about your question. We performed differential expression analysis for normal light group and clouded low group, such small groups. And here we have some top 15 high informative genes for separating these two groups. And you can try to plot all 52 samples with these 15 genes and look and compare with the previous 15 genes, which looks better.
So where do you find those files? Under output files, you have this folder.
At hands-on.
Oh, okay.
At hands-on folder.
Oh, okay, you have it here. So under hands-on, top 15 genes, VSEC2 out processed, and you can just use that file. And if you don't know how to deal with that file and what are the values inside, go to Transcriptomics 2, free course. It will explain what to do. Okay, now we're going to skip a section of our presentation because of the limited time, and we're going to go to the section dedicated to R. So one thing that we wanted to make available on the platform is continuity, potentially to other methods that people are using in the lab or other courses that you're taking, coding and visualization. And what we did, and you can see in this little video, is after you complete a pipeline, you probably notice that there's a zip file called R-pack. So for example, PCA will have a whole zip file called PCA R-pack.
Okay, now let's go to the folder called 5 Platform R scripts. Here we prepared 4 scripts for dealing with PCA, H clustering, or SVM, and stepwise LDA. Today, let us consider PCA. Yeah, great. We can open it with it. Okay, and here we see the code. Each file starts with needed libraries, so you can install them in order to run this. And that's all. We have TCA plot.pdf, and it already, yeah, and we see it, but just one click, and script works. Okay, let's go through it. we see options from the platform. Remember, we were selecting something like transpose, center, scale, how many principal components to draw in the web interface. And here we have all these options in the code. So you just can modify input file, you can transpose the data or not, center the data, scale, and so on. So this is just simple script. Not so simple as if you write it by your own hands, but okay, so let's start. Just look by code chunks. First, we read the data. So it has a header. We have row names in first column. Yeah, and that's all. Then it's option to transpose the data. So we have done it. We have already done it for you. So you know, transposing the data in R sometimes it's not so simple because we have some text columns, text rows, and these all are already handled. Yeah. Then we assign some group names, and here's a main line calling PCA, just PRC function. It's from stats package already installed in the R built-in package. And then about drawing, you will see PDF, and if you are writing a manuscript or something else, perhaps you want a more beautiful plot than this is. Yeah. So you can modify this string, and we have already improved script in our code directory. Here we have PCA folder 2. It is a file for generating such pictures. So you see that we have percents of variation explained by the principal components. Here we see some group labels, and we have sample names instead of points. And let's see how we can modify our script to draw beautiful pictures. So you see that first rows are probably the same. We just load the data, just read the tables, transpose it. Here we transpose, assign numeric class to all expressions. If you want, we can go really through and just analyze each line. Do you want it, or just go to visualization section? Okay, let's see how we can visualize. In platform code, we use ggplot2 library, and here we use ggfortify library. It's just a package for aimed to PCA plots. And here we see how it works. The function is called autoplot. Here we pass result of PCA. So here I applied PCA, and I pass this object to the just as argument. Then I say from which column to take colors to our groups. Here I have some data frame called groups with column with group names, and I say that please take the colors from that data frame. Then I have option label is true, which just draws label names on this plot, and yeah, I can set label size, shape equal false. Let me set it to true and see what happens. If I will zoom a little, nothing happened. Shape. I need to remove the slide. Yeah, and now I removed shape equal false, and I see points here. So shape it's just an argument for drawing points. I think that they're not needed now. It's more beautiful without them. So we can... Okay, and then frame is just for drawing these ellipses here. And here we have option frame type. And just for practice, please remove it and look what happens. Just run this code by yourself. If you have some problems, please ask me. So if you opened RStudio first time in this folder, you need to copy input file to that. Okay. Remove it. And you see that we have polygons instead of ellipses. Okay, so and I have commented there are some other options which are perhaps hard to understand because there are some have mathematical meaning, but let us uncomment these strings and see what will happen. So are you following me and running that? Or... Okay, so these arrows are called loadings, and what does it mean? Let us imagine, for example, three-dimensional space and three axes. You know they are orthogonal to each other, and what actually PCA do? It selects one more set of axes, for example, just like this, and then make a projection to two of them, and then we can project initial axes too. So these axes are projections of initial axes, and our initial axis was gene names. Yep. So for each arrow we have gene name, and how to interpret this picture. For example, let's take this gene. We can see if we go by direction of this arrow, then expression of these genes is growing. So, and we see that these two groups are located, these two groups and this group are located just on the line of this arrow. So it follows then big values of this gene expression are associated with these two groups, and small values with this group. So we can just go by these arrows and see how the gene expression affects the separation of the groups. So it is really important. And we see that some genes are not important, just short arrows.
So is that clear?
Yeah.
It wasn't very clear to me. So if I look at this picture and I see N is G, 65618.
Yeah.
As opposed to N is G, yeah, that one. So what can I know about these two genes?
You can know that big expression values of these genes mainly correspond to this group, because arrow goes here, and small values correspond to these two groups. So it's yeah. So that's a projection of initial axis. So it's really makes sense to use. Yeah, and most long arrow is the most informative gene in our case. Okay. Yeah.
Why there is nothing associated with the function?
They are.
So direction is not necessarily with the
Yeah, just direction. We have these ones. It's just a combination. It's hard to imagine, which means projection from 15-dimensional space to the plane with some huge combination of axes. Okay, and that's about PCA. Now we can try to remember we created a heat map, and we've seen that to obtain much information, we need to replace columns just by colors, and this is really difficult to do manually. And we can use heat map in R, and it automatically does this column replacement, and we will see a really beautiful heat map, which indicates that this is the first group, second. We have a code for it in R code direction. Here's a heat map folder, and we can open it. Let me close RStudio and reopen it in this directory. Studio. Okay, the start is just pretty same that in PCA. We just load the data and transpose, because all machine learning methods actually wants to sample be row names, not column names. But for biology, it's more convenient to use it by column, so we need to transpose them each time. Okay, and here I have a huge heat map call, and let's reduce it and step by step append some options and see what they actually mean. So let me. Oh, okay. First you can see it. Oh, no, we can see it. One moment. I need to rerun. Let's install this library. Okay. Now let's see PDF. Now that is, you see power of R, just compare it with Excel plot. Here we have some hierarchical clustering, which orders these rows. And here we see just some groups that they are all red. Here we see green, and just they are all sorted in proper way. So let us see how it works, And which options do we have? Okay. Let's copy it, paste here, and command this one, and remove all options from here. So calling heat map from scratch in R. Okay, and that's how it looks.
Can you again show which places you commented at the beginning?
Okay, sure. I've just, you can just remove all options from this call, just leave expressions. Okay. Where's my PDF? And here how row heat map calling looks like. Well, let's look at the differences. Should I open it one more time? Okay. So first we have some density plot on the color key. That means that red values are used more frequently than these yellow values. Yeah. So we have a lot of red cells in our heat map. But actually, people don't like this, and we want to remove it. So for that, we can use density information node. Put it here, rerun, and see no density information here. Now about these strange lines here, strange traces. What is that? At each column, we have some dashed line, which indicates the mean expression level. Here is it, just orange color. And if our value is lower than mean level, then solid line is drawn at left of this dashed line. And if it is yellow, it is drawn at the right. So I don't know why it should be used and why it is the default option in heatmap 2 function. So every time you want to draw heatmap, first of all, you put these two options here just to prevent traces and density info. And then, okay, and then our heat map is more clear now. So what we want then? Then we see that our gene names are not fitting in our PDF. They are truncated here. So we can add some margins. For example, 12, 12. You can modify these values as you want. And here we see that now they are really fitted here. Okay, and then we see that row names, sample names are covering each other, and we want to reduce font size. For that, we have option CX row. So 0.5 is a percent related to the initial font scale. So when I type 0.5, it means please add font size 2 times less than initial one. So when we run it, we see that they are not overlapping here, and this is great. And the most challenging part is then to mark groups in this plot, because we know that H clustering, it's just hierarchical clustering here, it reordered our rows, and we see some groups here. For example, we see yellow there, red there, long red there, and we think that this reordering actually matches with our groups, our 4 clinical types, and we want to add some colors to indicate which group is where. For it, there's an option called row side colors, in which you need to pass an array of colors associated with each of the group names. So for that, we should prepare this array first. So we have an array called groups. Let us print it and see what it is. Yeah, here's group. Group is just an array. Top line is a header with group names, and we want to convert them into the colors. For that, we initialize an empty array of row colors, iterate through groups, and just some ifs. If group is luminal, then color is cyan, normal like orange, and so on. And after that, we append this color to the row colors array just by calling C. Do you know what C means in R? C is just for creating an arrays. So we just concatenate to arrays by this line. And then we can pass this option to the heat map and see the thing that which is not possible to do in Excel. You see that this is a luminal group, and I don't remember group names and their colors, but
okay, but that actually is the point. So, as you remember in the previous session, we wanted to look at the differential expression, or sort of the difference in expression between the normal line and the cloud and low. So could you make all other colors white and just make cloud and low red and normal light green? Can you make this heat map with those colors? So we just look at the difference between cloud and low and normal light.
question here. Yeah, that's for you now. Suit. Can you reopen the RStudio, close and no, you should open it, go to the folder with file. Yeah. Close it now. And open that. Yeah, RStudio. And I'm trying to see this. Yeah, source. Okay, you need to install these. Install that packages.
So Kevon, can you just quickly show how to change the colors for powder, no, and normal?
Just here?
Yeah. Just so we see the same thing.
Okay, just white. Yep. And this is.
Okay, so something like that instead of red and green, but that's the idea, right? That's what you got. So why is this useful to have this information? Any idea how you could use this functionality if you were studying these genes and trying to understand about these samples?
It works. I think it
So
works
any
now.
ideas how this could
Just the
work?
warning.
So what's highly expressed? Can you tell me? Yeah. Right, but what's interesting, yeah, you rightly point out, is that this little section is unique to this group, right? Right away, it kind of jumps out at you. Anything else that you see in this example? Exactly. So which gene is this? Yeah. Okay, so what we found out in this little exercise is that we have a heat map that is more easy to work with than Excel, because right away we say we can group together, right? And then we also can quickly find genes that are maybe of interest based on their expression value. So, yes.
Yeah, then one more thing there. That remember we set linkage type toward dot. Let us see now. Yeah, let me run this. We see that there are some kind of not perfect reordering. You see that this group is splitted by orange group, and there are two luminal types. So ward.d2 is just another modification of the algorithm, which affects to the way how distances between clusters are calculated. And if we append this long line here, not here, it will use this linkage type for dendrograms, and here you see better clustering. And here we see, for example, an outlier. Here is it. And it's actually luminal, but it's more similar to, I don't know which group it is. Okay. Not the purple. Basal. I'm not good at colors, so I don't always remember.
What is anything online? So are there any questions about this so far? So since we have quite a lot of time, yeah, so let me go through differential expression and then you can show them the assignments, okay? So you can show them the assignments, PDX and TCGA, and explain.
Or I can go through differential expression if you want.
Okay, well, let me go through that. I just don't want to spend too much time.
Okay.
So let me just go through it. But if you can explain the assignments and the files and all of that. Okay, so the thing that we would like to finish with, and this is a little bit different than
the logic, I think, of what we typically would expect in a progression. But up until now, to summarize, what we have is this. Okay, let me go to the beginning here. I just want to do a little summary of what we have done so far. Okay, so we saw that there's a basic challenge in working with raw data. It's hard to interpret and it's hard to navigate even the process data because it has so many different genes. Now in RNA-seq, there's a few ways to look at the differences between samples. We either can have a simple separation of just two groups, or we can have multiple types of groups and classes, and then we need to apply more advanced methods. So in this session, we looked at transcriptomics processing, RNA-seq processing section. We went through how to do a typical expression table with pre-processing and quantification. And then we looked at data mining to look at unsupervised methods where we just look at the groupings within the data or patterns within the data, and then use that pattern to train and classify a new sample, for example, a group of samples. This section we haven't touched on, but it's very useful. So you can see here the pathway with different colors, right? And so when we look at differential gene expression, this is a very useful thing to do because we can annotate pathways and we actually can see the expression of genes on those pathways. So let's take a look back at the RNA-seq pipeline. So we'll open back this section, okay, and we'll quickly create a differential expression pipeline. And in the meantime, talk about how it's useful. So in our little exercise here, in our exercise, we saw a pretty clear difference between cloud and low and normal light. So if we can take, again, those two groups and create the same pipeline that you see over here. So what are we going to do? We're going to do pre-processing again. So Trim-O-Matic to remove the adapters, PCR clean to remove the duplicates. Then we're going to map it on transcripts. So we're not going to be looking for new isoforms or anything like that. We're going to generate an expression table, and then from there we're going to use dsec2 or R or edgeR to do differential gene expression. Okay, so let's look at the files.
We need to specify groups in this pipeline.
Yeah, so let's first find the files.
Same SVL.
Okay, yeah. So in our input files, expression quantification, we have the samples SVL. So as you remember, on top we specify pair ends, and over here we specify human genome 38, upload files and groups, sorry, URL bulk upload, input files, expression quantification samples SVL, and now we can define groups. So we'll have two groups. So yes, how many we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 11. So 11 samples total. The bottom 5 are going to be in group 1 or group B, and the rest in group A. So if you just drag them like this, it worked? Okay, so you should see something like this. You have two groups, 6 samples on one, 5 samples on the other. All right, continue. Here we have something for segmentation. It's not going to be used here, but just do select all, submit all changes, and then we can continue. So start, Trimmomatic, PCR clean, bowtie 2T, RSM expression table. And then whichever one you want, I'll use dsec2, end. Okay, then you would click on Run Pipeline. Again, it's not necessary for us right now. Let's just take a look at the output files. Okay, so yeah, let's go to the YZNA, just in principle, do you know?
I think some amazing data. Yeah, you can click on no sizes.
Okay, so this is a little bit more clear, right? So we have false discovery rate. It's already sorted for us. So we see that above a certain threshold, right? So we can select, for example, PAM50, right? So PAM50 is a 50 gene panel, right? So we can just take some number, like let's say 50, and just select the top 50. Or we can try and work with the values, right? And here we have FDR is basically the p-value adjusted for the total number that we have. And we can set some threshold. For example, if I just do the first option, so let's just take the first 50, I will have 50 gene names, again, an ensemble ID that I will want to use for annotation. So then I want to see what types of differentially expressed genes, so not only differentially expressed individual genes, but what pathways they're in. So quickly, if I take this table that I got over here, and I go to areas of analysis, utilities, and I use this EdgeR table, okay, so here I have this EdgeR table. And over here I have this button called Human Gauge. So the Human Gauge, again, gives us a lot of options here, but basically let's look at a few things over here. So what types of outputs are we going to be interested in, right? And so we saw the table, so we saw this FDR column over there, right? So we can use this threshold to make it either less genes or more genes. Over here, we can look also what type of KEGG database we will use, right? So we have, a few options over here. Okay, and then we can look either at down-regulated, up-regulated, or combined options. And we'll see the output here in a second. Okay, so we see column name header is present, where the gene IDs are, and then where are the actual expression values, right? So let's take a look. Okay, so here we have EdgeR, yeah. So again, over here, let's just, if you want to go back in the platform, you just do a right click. Okay, so I can select it again. So where is my differential expression measure, right? So here it's in column 2. Let's see, where is it in the other one? Okay, so here you see it's column 3. So we want to modify that. If you used dsec2, you would want to modify that. Let's do save. And run pipeline. Now, in the meantime, let's take a look at the output. So output files, post-processing, and then you have gauge. And so under gauge, you will have many PNG files like this, and you can probably recognize the KEGG pathways. But also down here, you have a table. And so here again, you have these different pathway names, and you can now find them. So let's take this first one that's called ribosome, right? And we can see it's number 03010. So it's 030, this one. So what's nice about this is that we can see which, I don't know what ribosome pathway necessarily means, right? But if we take this name, so if we go back to the table, right now I can Google this. Okay, and this is the full pathway. So I can see all of the genes that are involved in this pathway, and this is basically the picture that we saw over there. So the idea behind this is that if we have differential expression of genes between just two groups, we can then use those values to generate enrichment analysis with pathways and with annotated genes, and we can look through those genes and we can look through those pathways and we can order them by significance. So amount of genes, so if we quickly look at here again, you have p-value, q-value, and set size. So set size is the number of genes in that pathway.